All right, welcome back to our bacteria, viruses, and prions lecture. I'm not quite sure why the other one stopped so early, but let's continue. So bacteria, here we had left off talking about how to name them properly, making sure you capitalize the genus, lowercase the species. And sometimes the name tells you a little bit about the organism. For instance, maybe who found it or something descriptive about it. For instance, most of you have heard of strep throat, and that actually tells you that it's Streptococcus. So with that, bacteria have different shapes, and that's shown in the big picture here. Most common are the cocci, the bacilli, and then there's some other irregular shapes. And shapes were the first way we started characterizing by looking at microbes under the microscope. Remember that the shape and the morphology, so how they're arranged, is based on cell division. So Streptobacillus grows, divides, but stays connected, versus single ones grow, divide, and then release from each other, so they're in singles. The shape's affected by the cell wall, and here you see some of the less common, more kind of bent or spiral bacteria, and many of them are pathogenic. Binary fission is the method of replication for bacteria. So again, they grow, double in size, double their chromosomes, split in half. And like I said, sometimes they'll stay together and other times they grow separately. Because cell division is such an important component and bacteria do it quite quickly, some can divide every 20 minutes. This is a major target for antibiotics. So, for instance, DNA replication. Bacteria use some slightly different proteins than we do, so we can target drugs to block DNA replication. The cell wall. So forming this outside coat, which we're going to talk about in a second, is a major source of antibiotics, and as well as prokaryotic ribosomes. So remember, for any pathogen, you want to find something that is unique to the pathogen and not in common with the host. So again, something unique about bacterial cells as compared to eukaryotic cells is that they have this outer cell wall made of a component called peptidoglycan, which means peptides and sugars, or amino acids and sugars. And there are gram-positive bacteria that have thick peptidoglycan cell walls, you can see right here, and gram-negative bacteria that have a much, much thinner peptidoglycan layer, and then they actually also have an outer membrane, so another phospholipid bilayer. And there's a staining technique that you used in microbiology called the gram stain, and gram-positive and gram-negative will stain differently, and this is an important characteristic of the bacteria because like I said, many antibiotics target cell wall synthesis, and they're going to affect gram-positive different than gram-negative. So we'll have antibiotics that are specific for gram-positive and antibiotics that are specific for gram-negative. Something else to note about gram-negative is the lipopolysaccharide, or LPS, that some express on the outer membrane. And LPS is considered an endotoxin. So this is something that can cause pathology or disease in people. Mycobacteria, on the other hand, are slightly different. They are neither gram-positive or gram-negative because they actually have, this waxy outer layer. And this waxy outer layer gives them resistance to drying out.